So good morning, students. Good morning. Today we'll discuss about structural variations. So we are discussing chromosomal mutations, which we can also call them as chromosomal aberrations. Among them, there are two types: changes in number and the changes in the structure. So changes in the number comprising of euploidy and aneuploidy we have already completed. Now we'll start structural variations. As the name implies, these mutations bring about a change in the chromosome structure. And this change in the chromosome structure obviously results from a breakage. It results from breakage, which can be caused by either X-rays, various chemicals, and even naturally. That means spontaneously. But broken segments will never get dissolved. They always tend to rejoin. When you take a chromosome, okay, each chromosome has telomeres on the ends. What is the function of telomeres? The telomeres function, as the scientists say, is to prevent fusion of two different chromosomes. To prevent fusion of two different chromosomes is the function of the telomere. When this chromosome is broken, so we now see the broken ends. So this is a broken end and this is a broken end. These broken ends, as they do not have telomeres, they can rejoin. So that is the main principle behind breaking and rejoining. If there is more than one break, rejoining occurs randomly and not necessarily with the same correct ends. That means it can rejoin with another chromosome also, or with the chromosome at a different location. We cannot say precisely what actually is going to happen. So these changes that result in chromosomes due to breakage and rejoining constitute structural changes. There are four types of these structural variations. One is deletion, also called as deficiency, duplication or repeating of segment of chromosome, inversion, and translocation. When we were dealing with euploidy and aneuploidy, aneuploidy results in the reduction of the number of chromosomes or gain in the number of single chromosomes, like hypoploidy and hyperploidy, which are not significant in evolution. Why? Because we already have seen certain disorders related to these. For monosomy, there is Turner syndrome. For trisomy, there are so many: Downs, Edwards, Patau, Klinefelter, etc. Thus, they are not significant in evolution. Whereas in euploidy, there is a mechanism called as polyploidy, which brings about what we call a change in the number of sets of chromosomes that is significant in evolution. Likewise, here also, when we deal with structural variations, deletions and duplication to some extent in general are not that much significant in the formation of a new species, whereas inversions and translocations are found to be significant in evolution in the form of a new species, in the formation of a new species. So inversion and translocation among many are said to be significant in evolution. Why? They are involved in the formation of a new species. These structural variations, whether it is deletion, duplication, inversion or translocation, we call them as structural homozygotes or structural heterozygotes. That means a deletion can be homozygote or heterozygote, and so on, all these different three. Coming to the structural homozygote, here both chromosomes are involved in the structural change. For example, this is a pair of chromosomes, first or second or third pair, first chromosomal pair. Okay, what happened is, here there occurred one break. If the same break occurs in the other chromosome of the same pair also, then we call it as a homozygote. If it occurs in only one, if it occurs in only one chromosome of this pair, we call it as a heterozygote. So the structural variations can be homozygotes or heterozygotes. Deletion homozygote, deletion heterozygote, duplication homozygote, duplication heterozygotes, and so on. In structural homozygote, both chromosomes of a pair of homologous chromosomes are involved in that particular variation. In heterozygote, out of this pair, only one shows the structural change. And these variations, deletion, duplication, inversion and translocation, can be classified into two types, where deletion and duplication result in bringing about changes in number of genes. They bring about a change in the number of genes. Here, deletion results in change in the number of genes by loss of genes, and duplication results in change in the number of genes by addition of genes. And coming to the inversion and translocation, they are nothing but changes in gene arrangement. There is neither loss nor gain with respect to a particular chromosome, except in certain cases which we will discuss later. Here just there is a reorder, gene arrangement, reorder of the genes takes place. Here this is 180 degrees. So when rotation of a group of genes through 180 degrees within one chromosome results in an inverted portion of the same segment, that is called as inversion. Thus just there is ABC, DEF normally, now it will become ABC, FED. So there is a rotation through 180 degrees resulting in inversion of the chromosomal segment. So here there is neither loss nor addition. Same genes are present, but there is a reverse order. Okay, and here we call this as inversion. Likewise, exchange of parts between chromosomes of different pairs is called as translocation. So there is another chromosome, LMNOPQR. Okay, now what happens? This DEF is changed to this one, and OPQ comes here. What happens now? The new combination will be LMNOPQ, ABC, sorry, element DEF and ABC OPQ. So here also there is generally no loss or addition. What happens? There is just a gene arrangement. There are some what we call exceptions for this, which you will learn as you go through this topic. Coming to the examples of all these, let us consider a small chromosome with about these 9 different genes on different loci. First locus has small a, second locus has small b, and thus there are different genes on different loci of this chromosome. When deletion happens, what actually happens here is, when deletion occurs, ABCGHI remain. That means DEF, this segment is broken. So in this part, in this chromosome, this part where DEF is said to be present, it is said to be lost, and these two segments rejoin. And this involves two breaks here, one break here and the other break here, followed by rejoining. And such a structural variation where certain genes are lost due to deletion constitute deletion mutations. And in another case, duplication, a part of the chromosome is duplicated. That means this DEF is again repeated, either at the same place or at different place it can happen. So when a segment of chromosome with a few number of genes is said to be repeated, this we call it as duplication. And when there occurs a rotation through 180 degrees followed by breakage at here and this location, followed by rejoining at these points, it results in a chromosome with this order. So where previously it is ABCDEFGHI, now this is in reverse order, ABC, FED, ABC. Now DEF, as it is rotated, it comes to like this, and joining takes place between C and F and D and G, and thus this type of structural change is called as inversion. Okay. And coming to the fourth one, translocation. Here two non-homologous chromosomes are involved. Why we are saying this so? If a pair of homologous chromosomes exchange between them the same portion, we call this as crossing over. Okay. Even if it is a mutation also, there is nothing that changes except the alternate forms of alleles are changed. There is a new combination will be formed. But when one part of this chromosome is breaking here and this is exchanged for this one with the other non-homologous chromosome, then we call it as translocation. Okay. So thus there will be, this is the one chromosome, this is another chromosome, and there is exchange of segments between these two chromosomes resulting in a structural change called as translocation. Okay. So as you have heard about all this, in the given slide, can you identify which one is deletion, deficiency, duplication or translocation? Okay. Who is going to tell? I will give numbers 1, 2, 3 and 4.
So the 3 duplication.
3 duplication. Why?
2 times
CD is repeated, hence it is called as duplication. Very good, excellent. Second one is translocation. Why? So this is from a non-homologous chromosome adding to this one. Okay. One is inversion. Why? Very good. Next. Here it is deletion. Very good, excellent. Now let us deal with one by one. Deletion or deficiency. We can define this as loss of Deletion always is a loss. Loss of a chromosome segment is called as deletion or deficiency. It is of two types. One is called as terminal deletion, the other is called as intercalary or interstitial deletion. So let us say this is one chromosome, A, B, C, D, E, F, G. Now what happened is, there is a break here. Why this break occurs? It may be due to chemicals or X-rays or anything. So when this occurs, we come across with two parts. One up to A, B, C, D, E, F and the other with G. Okay. So here there are two parts. One is said to be, we are saying it is loss. Why we are saying it is loss? Because of the fact that when a chromosome is broken, let us say it is broken here. It can, this part can rejoin with another one, or it is completely lost. Why? Nucleases are the enzymes which break down DNA and RNA. So DNA is always broken down by DNA is. The protection for a chromosome from nucleases is, as far as I know, lies in the centromere. So the chromosome which does not have a centromere is recognized by DNase and that is digested. So that now we will be left with this chromosome where this part is said to be removed or deleted and digested, and so that there is no representation of whatever the genes are present here. For example, there is a gene for A and B, one for insulin, one for growth hormone. If that is so, we cannot have those two genes for those two hormones. Okay, so that is how deletion is said to be dangerous. If the one break removes a part of the chromosome from one end of the chromosome, it is called as terminal deletion. In another case, there is a chromosome A, B, C, D, E, F, G, H, I, J. Here what happened is, a break occurred here between BC and DE. So that what happens? CD is said to be removed. Now, after rejoining, there are two breaks and rejoining. So rejoining of A, B, E, F, G, H, I, J. So there is shortening. The chromosome which is this much long, due to the loss of CD segment, the length decreases. It involves two breaks, one here and the other here, followed by rejoining between these two. And such a deletion is called as intercalary or interstitial deletion. So let us see who discovered it first. Deletion was the first structural aberration or structural change or structural mutation or structural chromosomal mutation detected by Bridges in 1917 from his genetic studies on X chromosome of Drosophila. Can you tell me with what C.B. Bridges is related to? In the previous class we have already discussed about it. C.B. Bridges is related to what? Very good, excellent. Genic balance theory, not genetic imbalance. That is different phenomenon. You have come closer. Genic balance theory, where sex index X by A ratio determines the sex in Drosophila. So while doing those experiments, he discovered deletion in Drosophila maybe. Stadler is another scientist who produced breaks in the chromosomes of the plants by the help of ionizing radiation such as X-rays. So we are saying that X-rays, chemicals and some other agents can create deletions. In order to prove it, Stadler exposed certain plants to the X-rays and found deletions. Next, coming to the deletion homozygote and deletion heterozygote. Deletion homozygote is always lethal. Why? Because in homozygote, if this is, if this is a pair of chromosomes where due to deletion this part is lost and this part is lost. That means whatever the genes present here and here are not represented on both the homologous chromosomes. Thus it is a deletion homozygote. So this neither a single gene is said to be present with respect to the characters that are determined by these two segments. Thus it is said to be very lethal. Whereas if one is said to be lost and the other remains, this part remains, that means at least one gene for each of these characters are represented, though this portion is said to be lost. So thus a smaller deletion in heterozygote, that not even bigger deletion, smaller deletion in heterozygote can be tolerated when compared with the deletion homozygote. Okay. Now, how do you identify a deletion mutation that has happened in a cell? So when we observe the cell during meiosis in zygote, what happens? Can anyone tell me what happens in meiosis, zygote of meiosis? Synopsis. What is synopsis? Pairing of what chromosomes? Very good. Pairing of homologous chromosomes. So this is the first pair of chromosomes where pairing should take place. Now, out of these, one is said to be exhibiting deletion. This is the chromosome which shows deletion, and this is normal. Now what happens? How the pairing takes place? This portion does not match this one. So when we consider such a mutation, let us see, there is an intercalary deletion. There is an intercalary deletion with a specific segment of this is lost and this is present here. So A, B, C, D lost, E, F, G. So here this is normal. A, B, C, A, B, C, D, E, F, G. When such thing happens, when these two chromosomes come together, this is A, B, E, F, G, and this chromosome up to AB it is normal, up to EFG it is normal. In order to compensate for the loss of CD, here in order to match this during pairing, it undergoes looping so that pairing between these regions can easily take place, and this we call it as deletion loop. So when you observe deletion loops during zygote of meiosis, you can, when you observe these loops, you can say that some structural change might have happened. Okay. and such loops are called as deletion loops, by which we can identify a structural change. And you have to remember that this deletion loop occurs not in the chromosome where deletion has taken place, but in the chromosome which is normal. So deletion loop is always seen in a normal chromosome, but not in the chromosome where the structural mutation has taken place. Clear? Any doubt? Good. Let us continue with deletion. So deletions generally produce genetic and physiological effects. When homozygous, when it is a deletion homozygote, most of the deletions are lethal, as we have said before. That means they decrease the viability of the organism and the organism may or may not survive. Why? Because most genes are necessary for life, and a homozygous deletion would have 0 copies of some genes. Thus it becomes lethal. When heterozygous, that means a deletion heterozygote, the genes on the normal homologue are hemizygous. So at least this portion AB is present, though AB here is said to be lost. It is said to be neither homozygous nor heterozygous, it is half zygous, hemizygous. That means there is at least one copy of these genes present. So when compared with homozygote, heterozygotes are tolerable. And here also in the deleted segment, so this is the deleted one, and this whenever a break occurs here during pack18, there must be a crossing over taking place. But as there is no segment here on the counterpart, there cannot be any crossing over. So crossing over is absent in deleted region of a chromosome, since there is only one copy present in deletion heterozygotes. We cannot think about crossing over in deletion homozygotes because that portion is already lost. Crossing over may take place at any other place other than where the deletion has taken place. In Drosophila, several deficiencies induced the mutants like blonde, pale, beaded, curved, notch wings, minute wings, etc. So many such deletions resulted in numerous phenotypic characters in Drosophila. And coming to the examples for deletions in humans and their effects. Deletions are lethal, as we have said, even as heterozygotes in human beings. Why? Because they result in zygotic loss. So meiotic pairing may or may not take place. If it does not take place, what happens? Gametes are not formed. If meiotic pairing takes place and leads to the formation of gamete, and this gamete is said to be gets fertilized, even then after the formation of zygote, because there is no such gene for certain characteristics due to deletion, the zygote may be lost. Even if the zygote continues the embryonic development, the embryo may aborted. The development of the embryo may be aborted. Or even if what we call lives, the infant may die. Even if the infant continues to live, it leads to certain disorders. So this is the cascade of effects of deletion in human beings. So sometimes thus infants with small chromosome deficiencies survive long enough to permit the abnormal phenotype to be expressed. And one such we are going to see is Raynaud syndrome in human beings. It is also called as cat cry syndrome. That means when the person with this syndrome, when he speaks or cries, means when he shouts or speaks, it resembles the sound of a cat. Hence it is called as a cat cry syndrome. It was described by the first time by Lejeune and other scientists. Et al. Means and his colleagues. If there are more than two, if there are only two scientists, Beadle and Tatum, Watson and Crick, we can name them two. If there are more than two or three, then we use the word et al. Lejeune et al. Means Lejeune and three or four other scientist colleagues. They described it in 1963. So the karyotype of this will be 46XX. This is female and like this. If it is male, this is like this. So what does this show? Here 5 refers to fifth chromosome, P refers to small arm. Fifth chromosome is a submetacentric chromosome with a small arm being called as small p and a long arm called as small q. So remember, short arm in a submetacentric chromosome is always designated by small p, and long arm in a submetacentric chromosome is always represented by small q. Now what happened? Deletion occurred here, where in the small p chromosome. So we call this as 5p deletion. So there is a segment on the short arm of fifth chromosome is lost, and it results in certain characteristics like an IQ which is less than 20, and malformation in the larynx leading to the cat cry, a face which is moon face, saddle shaped nose, micrognathia. Micro means small, gnathos means jaws. Small jaws are mandibles, and low set ears, microcephaly, small head. So that means you can see from this portion, from this characteristic, that the short arm of the fifth chromosome where the deletion has taken place, this is responsible for a number of characteristics. First one, this is having a gene for IQ. Second one, this is a gene for larynx. Third one, there is a gene for shape of the face. There is a gene for shape of the nose, for jaws, for ears, and for the size of the head. So thus this last portion not only results in loss of one character, but it is manifested in the loss of too many characteristics, because as many genes are present in this lost segment, as many characters are said to be found to be deleted. Okay, if it is said to be homozygous deleted, deletion homozygote, what happens? Neither small head nor large head, complete head is absent, maybe like that. A deletion heterozygote is said to be very lethal. And coming to the other examples, deletions were also observed in prokaryotes like Escherichia coli, T4 phase, and also lambda phase. In Escherichia coli, deletions of up to 1% of the bacterial chromosome are observed by scientists. That means large number of deletions may take place in prokaryotes. In lambda phase, 20% of the genome is found to be exhibiting deletions. So thus prokaryotes are more vulnerable for deletions. And other examples other than cat cry syndrome, we see notch-wing mutation in Drosophila, which is a recessive lethal gene occurring due to deletion. Another example is pseudodominance. Pseudodominance. This can be exemplified by a waltzing mice described for the first time by Gates in 1921. What is a waltzing mice? You might have heard the movie song Dil Hai Chota Sa, isn't it? From Roja movie. In Telugu we call it as Chinni Chinni Asha. Okay. So the entire song has only one beat of music. That beat is called as waltz beat. You can hear that. Ching, 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 like that. The same beat goes on from starting to ending. That means here, this mice repeats the same behavior. The same erratic behavior is repeated continuously by the mice. For example, if it is kept in a cage, if it moves from north to south, one time, that same thing is repeated continuously. Okay, in that case we call such mice are waltzing mice. Why this happens? Let us say there is a gene for normal behavior. There is a gene for normal behavior which is dominant, and this got mutated to result in waltzing mice. Okay, waltzing behavior. So this is recessive, this is dominant. So there is every chance for three different genotypes, capital C, which will be normal, capital C, small c, which will be normal but heterozygote, small c, which is homozygous waltzing recessive mice. Can you observe waltzing behavior in a heterozygote or homozygote? Dominant? Can anybody tell me? In heterozygous, can we observe waltzing behavior, capital C, small c?
No, sir.
No. Why? The dominant gene, normal gene, suppresses it. Okay. What happens if in the heterozygote this recessive gene is expressed, resulting in waltzing behavior? That we call it as pseudodominance. So what actually happens here is, this is a pair of chromosomes in mice where it is actually a heterozygote, and just a minute. Okay, where it is heterozygote. And now what happened is, a deletion mutation has occurred and this portion is lost. This portion of the chromosome is lost. That means the dominant gene is lost. What remained here? Only the recessive gene. Actually it is a heterozygote. Due to deletion, dominant gene is lost and only recessive gene remains. This recessive gene now results in the absence of the dominant gene, leads to waltzing behavior. Such a phenomenon where in a heterozygote, actually it is a heterozygote, here the dominant gene is absent, hence the recessive gene is expressed, and such a dominance is called as pseudodominance. It's a false dominance. A recessive allele of the normal homologous chromosome, due to deletion of dominant gene for normal behavior, it behaves like a dominant allele. That means it expresses itself. We don't say that it is a dominant allele. It is expressing. When we say that it is expressing, it is dominant expression. But when we are saying this is expressed in heterozygote condition, we can call this as like a dominant allele. But actually this is not happening in the presence of C. It is occurring in the absence of C, hence such an expression is called as pseudo-dominance. So in examination about Cry-Do-Chat syndrome and about pseudo-dominance, and notching mutation. These three examples may be asked for deletion or deficiency. And coming to the second one, duplication. Presence of a part of a chromosome in excess of the normal complement. That means extra part is said to be added. Then it is called as duplication. So deletion is loss, whereas duplication is addition. So some genes due to this are present in more than 2 doses. And coming to the duplication, let me explain the types first. Duplication types are four or five. 4 types. One is called as tandem duplication. Second one, reverse tandem duplication. Third one, displaced duplication, which is again of 2 types, homobranchial, heterobranchial. And fourth one, transposed duplication. So what is tandem duplication? In tandem duplication, this is the normal chromosome ABC, DEF, GH with centromere between C and D. And this DEF is said to be duplicated. How duplication takes place generally? Duplication may take place either due to unequal crossing over or due to misreading of the or repeated or during DNA duplication. When the DNA polymerase up to ABCDEF it reads and somewhat it reads and forms a DNA copy, and after that it leaves, and sometimes after some few seconds or milliseconds or nanoseconds, whatever it may be, after it rejoins and start duplicating the same segment. So that what happens, this same segment DF is repeated twice. And how it takes place due to unequal crossing over, we are going to see later. What I am going to say here is, in tandem duplication, the newly duplicated segment is present by the side of the original segment. It is the original segment, this is the duplicated segment. When the duplicated segment is present by the side of the original segment, it is called as tandem duplication. In reverse tandem duplication, the duplicated segment, this is the one, which is present by the side of the original segment but in reverse order. So here it is DEF, same order. Here it is FED, it is in a reverse order. Hence we call it is present by the side, hence it is tandem, but the order is reversed, hence it is called as reverse order. And the third one is displaced duplication. In displaced duplication, what happens? Here there is ABC, DEF, GH normal, and here this DEF is placed between G and H. Here the duplicated segment is not present by the side of the original segment but away from it is present. It is present away from this. In the second case also, the duplicated segment is present away from the original segment. The only difference between these two here is, in this case, it is present on the same side of centromere. This is the centromere and the duplicated segment is present on the same side of centromere. DEF is present on this side and the duplicated segment is also present on this side of the centromere. Hence we call this as homobranchial. If the duplicated segment is present on the other side of the centromere, this is the centromere, we call it as heterobranchial. So here in both these cases, the duplicated segment is displaced from the original segment, either on the same arm or on the different arm, based on which they are called as homobranchial and heterobranchial. And fourth one is called as transposed duplication. Here the duplicated chromosome attaches to a non-homologous chromosome. we cannot call this as translocation. Why? In translocation, there is no duplication involved. Though there is a transfer of one portion of segment from one chromosome to the other, like this, that segment in translocation is not duplicated. Here it is duplicated. So A, B, C, D, E, F, G, H. And here this DEF is duplicated, and this duplicated segment is transferred to another non-homologous chromosome, and hence this is called as transposed duplication. And last one is extra chromosomal duplication. There is a chromosome A, B, C, D, E, F, G, H, I, J. So here the portion between C, D, E and F, G is said to be duplicated. So C, D, E, F, G is duplicated. So here this is normal. There is no change here. But this portion, as it is duplicated, it has its own centromere. When it has its own centromere, it acts as an individual chromosome. Thus we come across with an extra chromosome due to duplication. So in the presence of centromere, the duplicated part of a chromosome acts as an independent chromosome. Hence we call this as extra chromosomal duplication. Let us see how this duplication originates. Origin of duplication involves one thing, that is breakage and reunion of chromosome segment with its homologous chromosome. As a result, one of the two homologous involved in the production of a duplication ends up with a deficiency, while the other has a duplication for the concerned segment. Another phenomenon that results in duplication is unequal crossing over. Unequal crossing over. It leads to exactly the same consequences for small chromosome segments. For example, in case of Drosophila, on X chromosome, there is a segment called as 16A. And this 16A gets duplicated, and this 16A, when gets duplicated, it can form a number of copies. Actually, 16A is responsible for the shape of the compound I. When this gets duplicated, what happens? The shape of the compound I becomes oblong and narrow. A normal compound I has a number of simple eyes. Each simple eye is represented in each of these squares, which we call them as facets. Below each facet has a simple eye. All these eyes put together constitute a compound eye. In a normal eye with the 16A configuration, on one chromosome one gene, another chromosome another gene in diploid, because it is diploid. Here the compound eye is normal. When this portion is duplicated, what happens? The shape becomes narrow and oblong. At the same time, the number of facets also decreases. So this is an example of unequal crossing over. How it takes place? Here you see this is 16A present on one chromosome and 16A present on its counterpart. This is a homologous pair. Crossing over should take place when it is equal. It takes place between the same segments. But here the portion of the X chromosome that is being crossed over is said to be just away from this, because the chromosomes did not align properly. It should have aligned properly like this, but it has aligned in a different way, somewhat leaving a small space, so that there is unequal crossing over, and this is added to this one. What happens now? This is lost. There is one deletion here and there is a duplication here. As I have said before, during origin, one chromosome due to unequal crossing over may result in deletion, the other is said to be having an addition of another copy. Thus, two copies result in due to unequal crossing over. If the same thing continues, three copies may also be added. So, what is the significance of this? Let us see duplication example. Bar I in Drosophila is an example for duplication. It results in narrower, oblong and bar-shaped eye with very few number of facets. This is described by C.B. Bridges again. It was determined by, that means the eye shape, what we call shape or shape of the eye is determined by shape of the eye and also number of facets is determined by a X-linked recessive allele present on 16A segment on X chromosome. When this segment is said to be on one chromosome, there is one 16A and the other 16A, that is normal, as seen here. This is normal. This is normal. Okay, when we place both at the same place. In normal, what happened? There are number of facets. Average is said to be 779. If due to unequal crossing over, duplication occurs on one side, we call this as heterozygous bar. Why? Because duplication has been on one chromosome of a pair and the other is said to be having only single one. So it is a heterozygous bar. The number of facets decreased. And duplication takes place on the other one also. Then it is a homozygous bar and the number of facets decreases. So this much is said to be the size of the normal compound eye, which gets narrowed down continuously. And if another is added here due to duplication, we call this as heterozygous ultra bar or double bar eye, and the number of facets still decrease to 45. If another one is added to this one, that means it is homozygous ultra bar double bar eye, and it still becomes very much oblong in shape and the number of facets decreases to 25. Okay, I'll post this slide in the group. You need not have to write down. You just try to understand. So this is how duplication results in a number of phenotypic expression in Drosophila. But here there is a change in the phenotype due to duplication. We call this as genetic divergence. But here genetic divergence only when it is resulting in the formation of a new species, we can say evolution has taken place. But here up to the level of genetic divergence only, duplication can bring about changes during the course of evolution. There is another example in human being, that is chromosome 3. A structural change in chromosome 3 results in a syndrome called as chromosome 3 deletion duplication syndrome. Chromosome 3 deletion duplication syndrome. That means one part is said to be exhibiting deficiency due to unequal crossing over, the other part is exhibiting duplication. Hence we call it as chromosome 3 deletion duplication syndrome. And we are coming to the end. The third one is said to be inversion. When a segment of chromosome is oriented in the reverse direction, even in duplication also there is reverse tandem duplication, but that is duplicated segment which is in reverse order by the side of the original. But here there is no duplication. Some genes or one segment of the chromosome with some genes in one order is said to be reversed, resulting in inversion. And the existence of inversion was first detected by Sturtevant and Plunkett in 1926. Who discovered or described inversion? Sturtevant and Plunkett. Inversion occurs when parts of chromosomes become detached, turn through 180 degrees and are rejoined in such a way that the genes are in reversed order. So this is a chromosome. A, B, C, D, E, F, G, H followed by centromere I, J, K. In this case, what is happening is a break occurs here between C and D because there is a 180 degrees orientation of this chromosome between gene C and gene H. Now, after the break occurs here, the broken segments join between C and G and D and H. So now the order will be like this: A, B, C, G, F, E, D followed by I, J, K. I, J, K, where centromere is present between H and I. And because of 180 degrees rotation followed by breakage and rejoining, it results in a segment which actually should be A, B, C, D, E, F, G, H, centromere I, J, K. Now it is formed into A, B, C, G, F, E, D, H, I, J, K, where this portion is in reverse order, reverse direction, due to what we call this phenomenon as inversion. And here in this inversion, inversion has taken place on one side of the centromere. On one side of the centromere, we call it as paracentric inversion. Paracentric inversion. If let us say there is no centromere here, there is no centromere ABC, sorry, this is got inverted. A, B, C, D, E, F, centromere is here, followed by G, H, I, J, K, L. Now what happens? When inversion takes place, the order will be A, B, C, D, H, G, centromere F, E, I, J, K, L. This is the order, final order, where the centromere is present between G and F. And the inverted segment is this one. So the inverted segment involves the centromere here. Then we call it as pericentric inversion. Coming to example, a certain segment may be broken in two places, as we have just seen. It may be broken here, and the breaks may be in close proximity because of chance loop. The breaks are said to be very close because there is a loop formation as the chromosome segment has rotated through 180 degrees. When they rejoin, the wrong ends may become connected. So these are the wrong ends, these are the wrong ends. These wrong ends are connected. The part on one side of the loop connects with broken end and different from the one which it was formerly connected. So formerly it is connected like this, now it is connected like this. This leaves the other two broken ends to become attached. So the part within the loop thus becomes turned around or inverted. It is of two types, pericentric and paracentric. Pericentric inversion includes the centromere as we have just discussed, and paracentric inversion occurs, the inverted segment does not include the centromere, it is away from the centromere. Hence it is called as paracentric. Both pericentric and paracentric inversions are significant for evolution. They are significant for evolution. So how they are significant for evolution? They bring about what we call reproductive isolation. They bring about reproductive isolation. So what actually paracentric or pericentric inversion which result in reproductive isolation we'll discuss when we'll deal with evolution. Okay. So this is pericentric and paracentric. One is pericentric, the other is paracentric. Okay. Which one is pericentric and which one is paracentric? Can anybody tell me? First one is pericentric. Why? Because the inverted segment includes the centromere. The second one is why? Because the inverted segment is away from the centromere. Very good, excellent. Coming to inversions in natural populations. In natural populations, pericentric inversions are much less frequent than paracentric inversions. That means paracentric inversions are more in populations than pericentric. So when these mutations occur naturally, spontaneously, with relation to inversion, paracentric inversions are more and pericentric inversions are less. In many species, however, pericentric inversions are relatively common. For example, grasshoppers. So in grasshoppers, though paracentric in general are said to be more in number, in grasshoppers, pericentric inversions are more in number. Okay, and paracentric inversions appear to be very frequent in Drosophila. So grasshopper, we see more pericentric inversions. In Drosophila, more paracentric inversions, but in general, more paracentric than pericentric inversions. Coming to the advantage of inversion, as I have just said now, fertility of inversion homozygotes and sterility of inversion heterozygotes lead to establishment of two groups or varieties which are mutually fertile but do not breed well with the rest of the species, which we call it as reproductive isolation. They are fertile but they do not mate with each other. In that case, the same individual, the same species is having two groups. Actually, all the individuals of species A should interbreed among themselves. Due to inversion, homozygotes and heterozygotes, what happened? Two groups are formed. These groups are fertile enough, but they do not mate with each other. So in due course of time, this may evolve into one species and this may evolve into another species. Thus, as they are reproductively isolated, they will evolve in different directions, leading to the formation of a new species. Thus, among all the three that we have so far discussed, deletion, duplication, inversion changes are said to be mainly responsible for or one of the reasons for evolution of a new species. And coming to the last one, translocation. It is nothing but the addition or integration of a chromosome segment into a non-homologous chromosome, never in a same chromosome. Then only we call it as translocation. It is of three types: simple, shift, and reciprocal translocation. What is simple translocation? In this case, terminal segment of a chromosome is integrated at one end of a non-homologous region. Okay, so this is one chromosome from the first one. This is another chromosome from the second one. So here this portion of this segment is said to be lost and this is added to a non-homologous one. Such one is called as simple translocation, where there is loss in one, addition to the other. But in total, there is no loss in the number of genes. Thus, there is a gene arrangement. Next, shift translocation. In shift translocation, an intercalary segment of a chromosome is integrated within non-homologous chromosome. So, this is again first chromosome and second chromosome. It is not deleted from the terminus, but this portion is said to be deleted from the middle of a chromosome and that gets translocated onto another non-homologous chromosome. Then we call it as shift translocation. And these are said to be more popular in Drosophila and Neurospora crassa. And coming to reciprocal translocation, here what happens? Both the first and second chromosome, which are non-homologous, they exchange their segments. Actually, exchange of segments takes place between non-sister chromatids of homologous chromosomes. But here there is exchange between non-homologous chromosomes. So this is called as crossing over. We call it as reciprocal transfer. Reciprocal means if you give something to me, I'll give something to you. These two chromosomes are exchanging their segments. Hence, we say that they are reciprocally transferred. Among all the different types of translocation mutations, there is one mutation called as Robertsonian translocation. So this Robertsonian translocation, according to scientists, is what resulted in chimpanzees evolving into humans. Chimpanzees evolving into humans. And I am leaving this class with two questions for you. One question is, we have seen examples for deletion, deficiency, and there is no example given for inversion. Okay, because there is no disorder so far noted in your books with respect to inversion. But there is a disorder related to translocation, that is Philadelphia chromosome. Philadelphia chromosome. So this is your first exercise. So you please go through the Google or books or ask your lecturers and post in the group what actually Philadelphia chromosome and which two chromosomes are involved in this and what kind of translocation is it, simple, shift or reciprocal. The second one is how chimpanzees evolved into humans due to Robertsonian translocation. So what is Robertsonian translocation? So these two are the exercises for you which we will discuss in the next class when we deal with gene mutations. Okay? Any doubts? Ask me, otherwise we will close. Thank you.